HSC, inheritance patterns in a population. So let's keep going so we can get to module 6. This is where we think and we consider how can a population's genetic patterns be predicted with any accuracy. So this is where we have two techniques we need to know when it comes to inheritance patterns. The first one is DNA sequencing. And how does that occur? With DNA sequencing, we begin by isolating DNA from cells. We get its sequential order of the nucleotides because that's what makes a DNA unique. It's those nucleotide pairings. And then we put in a computer, we process it, and we can then compare whole genomes of individuals. And we can also conduct transcription and translation of genes in silicio. And so, as a result of this, we get a lot of information. We get information surrounding single nucleotides and single nucleotides and their polymorphism. So if there has been changes in single nucleotides, we also get information about understanding that each individual has a different genome, and we can use that to then identify differences and similarities. Modern computation will also allow for patterns to be processed, and with those patterns we can then trace inheritance of genes, alleles, and also SNPs, so single nucleotide polymorphisms. So the change in a single nucleotide, which is then going to lead perhaps to a bigger change in the whole sequence. Wonderful. And then we've got DNA profiling. So DNA profiling, probably you might be familiar with it in one capacity or another. Definitely if you watch crime shows and thrillers, that's where you would have seen DNA profiling a lot. So this is where we collect samples from individuals. So common practice, for example, includes collecting blood, hair follicles, or mouth swabs. We digest that DNA. So basically what we're doing is, once we get that, we're going to cut up the DNA into small pieces using what we call a restriction enzyme, and that restriction enzyme will cut DNA in specific places where we want it to cut. And then once that happens, we are going to put it in this machine called gel electrophoresis. So in this, I'm still on my pen cursor, there we go. Let me go back. Okay, oh nice, I've got a laser pointer. Here we go. So we then have this machine called gel electrophoresis, right? And so in this process, gel electrophoresis, what we do is we put DNA here at the sort of the front of the machine here, and it goes from positive to negative. What happens is that we put in the DNA, we've got these samples, and we've got a marker that we use, and that DNA will then travel according to its what its constitution is. Usually we see that smaller pieces of DNA travel further in comparison to bigger parts of it. So anyway, so it travels from positive to negative, and then we're going to see travel. And using this, we can then compare this, for example, for fingerprints, and we can do that to compare fingerprints to sort of determine, to determine crime scene related suspects, to determine paternity. That's also where DNA profiling is used, to compare paternity and to determine if two people are related or not. So if you have a sample here, it's a crime scene, we have a sample of a hair follicle that was found on the crime scene, right? And we've got 3 suspects, we got their hair follicles, and now we have put it through, and essentially we can see that we want to see which ones match up to this initial, to the marker here, to the hair follicle that was found in the crime scene. And you can see that it's actually C that matches the most to the marker here. So whoever is C here in this scenario is the person who has who is responsible for that crime. Now, one thing to be mindful of with DNA profiling is that historically it has known to have lead to to be complacent, in the sense that there have been wrongful accusations, but for the you know, for the sake of the module, for the purpose of the module, we just have to know what the process is, and we just have to focus on what it is and how it occurs. So the degree of similarity equates to the degree of relatedness, and using this we can observe conserved regions of the DNA. So the conserved regions are the regions that are actually different between everyone. So we actually share a lot of the DNA, like a lot of our DNA is very similar, but we have conserved regions that then lead to our uniqueness. And it's also effective in humans because we have large stretches of junk DNA that we can use for this process. So remember I said earlier, not all DNA makes protein, and so we have junk DNA as well, which we can use for this sort of process. Now let's have a look at inheritance patterns. So this is what you're going to do. You're going to look at large-scale collaborative projects. You're going to look at how they have identified trends, patterns, and relationships, and how they have used that and applied that to have outcomes. So essentially, there are three processes that we're going to look at. Firstly, we have conservation management. So conservation genetics is the field of bio where essentially we combine molecular genetics, ecology, and biodiversity to propose species management. And it is used to, this process uses population genetics to identify any alleles that are at risk and proposes ways to preserve the biodiversity. And an example of a group that does that is the Australasian Wildlife Genomics Group, which is found in the Sydney University, and they have projects like the Devil Tools and Tech projects, the Koala Genome Project, and also the Marsupial Genomics. Okay, next up we have inheritance of disease. So we can actually utilize this data to look at the inheritance of diseases. So what we know is that statistically there is only, and it's crazy, there is only 0.8% of nucleotide variants among humans. So only 0.8% of a DNA is actually different to one another. And like I said, that's crazy. That means a lot of it is very, very similar. So what we know is that if diseases are arising, it actually would be because if diseases are arising and not everyone has the disease, but some people do, that means that lies in the 0.8% of variance, because it wouldn't be in the 99.2% which everyone has. It's going to be in the 0.8%. So we then utilize that idea to understand the cause of genetic diseases. Why is it? Is it due to random mutations that occur, or is it due to the interaction of alleles? And then we can use that to design therapies to repair genetic disorders. And a good example of this is the Human Genome Project. So the Human Genome Project collects collated data on thousands of genomes to identify genes, variable regions, haplotypes, and they have identified genetic causes of breast cancer, Alzheimer's, and also colon cancer. Alrighty, looking at human evolution. So finally, we know that cultural and regional groups are linked by the prevalence of particular haplotypes. So essentially that we're linked by these particular groups of similar genetic sequence, and we can map that to trace the movement and evolution of human species. And we've actually been able to do that to determine the origins of mitochondrial Eve, who we said that lived in Africa 20,000 years ago. And that is part of the International HapMap project, which uses human genome data to identify SNPs, haplotypes, and haplogroups, so groups essentially of similar genetic constitutions. And so from this information we can trace back lineage.